Dear students, today we start the unit 4. In the last unit, we had seen about the chick embryology. Now, in this unit, we had seen the general parts of the embryology. In this, the first part is the egg of mammal. As you see on the diagram, see on the screen, the egg of mammal. So, it is spherical in shape, microscopic, smaller size due to the absence of yolk. See, here the egg is very small, microscopic. But in case of the birds, reptiles, the eggs are big because their development is occur outside the body. But in this case of mammal, it is inside the body, so there is no need of yolk or albumin. Only the nucleus is required. So such a egg is said to be alethal or microlethal, very microscopic. And the egg is released from the ovary and it is surrounded by three membranes, namely corona radiata, a middle zona pellucida, and inner plasma membrane. See here in the diagram, it is shown. So, and there is fluid filled space between the zona pellucida and surface of egg, that is called as perivitelline space. So, this is the egg. Now, suppose the fertilization is occur. Then after fertilization, this egg, that this is single cell actually, now it starts to divide for the growth. So for the growth, the first requirement is the cleavage, means division of the cell. So that is called as cleavage. So the cleavage is holoblastic and unequal, means is divided into two. See here, I am showing this diagram. The A is a single zygote or cell is there. So there is no synchrony in the cleavage process. It is irregular and it proceeds as egg passes down the oviduct. So it in mammal it is inside. See this egg B diagram, two cell, then this is four. See this D, E, and lastly we get the a bunch of cell. So the bunch of cell we get here, and during this division the two types of cells are formed. The smaller one called as micromere and larger one is called as macromere. And then follow the macromeres divides into two, thus producing the 5 cell stage, and this is followed by division of other macromere into two and thus forming 6 cell stage. So here, see here, smaller cells and bigger cells. So E is the 5 cell stage and F is the morula. F is the morula, the bunch of cell is there. And a 7 cell stage, this is followed by division of the other macromeres to produce 8 cell stage, and the cleavage is going on further, and in all cleavages the macromeres divide first, and that is followed by the division of micromere, means first bigger cell and then smaller cell, and this results in the appearance of a 9 cell stage, a 10 cell stage, also so on, and the macromeres are more active in division. So this is the cleavage. Cleavage is nothing but the division of the cell. And ultimately the division of this all this thing is the next cell, that is called as morula, next stage. So see the diagram here, the morula, a cavity, a cleavage results in the production of the solid mass of cells, and this mass of cell is known as morula, and it has no cavity inside. It consists of two types of cells, micromere and macromere. But then they flatten and to form a covering, it is called as tropoblast. See the diagram left side, the covering cell or the trochoderm, and it is nutritive in function, and the interior of morula is occupied by the micromere, and there are inner cell mass. The inner cell mass alone develops into a body proper of the embryo. So the inner cell bunch is developed, see this one is developed into the body proper, that is the internal part of the animal. Now soon this process is going on. Soon, next, that is a blastocyst. Soon a cavity appears within the inner cell of mass morula, and this cavity is called as blastocele. See here, this is the blastocele in between. So a certain fluid is embedded into the cavity. As a result, the cavity gradually increases. As the blastocele reaches its maximum size, the inner cell mass is oriented towards the one side and is suspended from the tropoblast like a honeycomb. See the diagram. Honeycomb. And this side represents the animal pole. The other side is naturally vegetal pole. So the mammalian embryo in this stage is called as blastocyst. So this particular whatever you have seen in on the screen is a blastocyst embryo of the mammalian group, and this blastocyst corresponds to the blastula of lower animal. So we know that the fertilization, then cleavage, blastula, then gastrula. So in lower animal these stages are there. So same stage is here. So this stage in the mammals is called as blastula, or it is blastocyst. The blastocyst is in the form of vesicle containing a covering, that is trophoblast. See the diagram. Then blastocele cavity and inner cell mass attached to one side of the blastocyst. So inner cell mass develops into an embryo proper, and trophoblast cells which cover the inner cell mass are called as cells of Rauber. So this is trophoblast is also called as cells of Rauber. So in this way, this is the morula. Now, up till now, this development is taking place inside the oviduct. Now, the next process, that is implantation in uterus. Now, this embryo proceeds. Embryo means now we call as blastocyst. So, we proceed and the implantation is occur. So, implantation means the attachment of this embryo to the uterine wall. This is called as implantation. So, the embryo reaches the uterus in the blastocyst stage. Then it comes attached to the uterine wall, and the process by which blastocyst is attached to the uterine wall is called as implantation. Just I explain you. See how this embryo get involved in the uterine wall. So the implantation is really the adhesion of the trophoblast, the outer cells of the embryo, to the uterine wall. The adhesion occurs selectively where there is maternal blood vessels beneath the uterine epithelium, and by this attachment the maternal blood diffuses into the embryo. See here, this is the first diagram. That is, so the trophoblast cell, the outermost cells are getting involved with the attachment of the uterine wall cells, and the diffusion starts. The blood vessels are there and the maternal blood reaches to the developing embryo. So this is called as implantation. So A1 is the superficial implantation, B1 is the eccentric implantation, and C1 is called as interstitial implantation inside the uterine wall. So this type of implantations we can see when the embryo attaches to the uterine wall. So the first one, that is the central or superficial. So this type of implantation occur in to the uterus. The outer layer of trophoblast makes a superficial attachment to the uterine wall. This occurs in the pig, carnivorous monkeys. Okay. Then second one is the eccentric. So in the eccentric, what happened? Here the blastocyst remains in between the folds of uterine epithelium. See the diagram. See here, the in the eccentric, it is in between the uterine fold. So this type of implantation is occurring rat and squirrel. And third one is the interstitial. In this case, the blastocyst burrows deep into the uterine wall. So it goes inside the uterine wall. And examples are hedgehog, guinea pig, and some bats and man, etc. So these three types of implantation is occur in the mammalian group. So this is the egg of mammal. So we had seen from structure to the implantation. So the next process is the placenta. Now, once these blastocyst or egg of the mammal get attached with the uterine wall, so the trophoblast cell of the embryo and uterine wall, they get entangled or mixed together to form a one structure. That structure is called as placenta. Now, this placenta is responsible for the embryo development in the sense that they provide the blood, food, then exchange of gases are taking place. So, this process is continuous up to the parturition. So, next time we will see the placentation in mammals. Thank you.